Today's lecture will be dedicated to organs of special senses, namely the eye, the ear, and partly to the sense of the taste and to the sense of the olfactory perception. Eyes are highly developed photosensitive organs for analyzing the form, intensity, and color of light reflected from objects and providing the sense of sight, having attributed for this purpose mechanism of photoelectrical transduction. The eye has three tunics. Sclera and cornea is the outer fibrous tunic. The middle vascular glia or uva consists of the choroid, ciliary body, and iris, and the retina forms the inner tunic of the eye. The transparent cornea consists of anterior stratified squamous epithelium on Bowman's membrane, a thick vascular stroma, and an inner endothelium on the cement membrane. The white opaque sclera covers the posterior 5/6 of the orbit. It is composed of type 1 collagen fibers interlaced with elastic fibers. Here you have the general arrangement of fibrous tunic, vascular tunic, and retina, consisting of pigmented layer and neural layer, and this vascular tunica vasculosa consisting of iris, ciliary body, and choroid, and fibrous tunic consisting of the cornea and sclera. Here a more detailed diagram of the eye, which is having a lot of terminology. This topic is not so hard to understand that it is hard to reconnect the majority of the terms which are related to the eyeball. Here we have the cornea, sclera, choroidea, corpus ciliaris, iris, the retina, including its optic part of the retina and non-optic or non-visual part of the retina covering the posterior surface of the ciliary body and iris. There is the lens, which is focusing the light rays on the optic retina. The corpus vitreum. There are the anterior chamber covering the space being filled with the aqueous humor, covering the space between the posterior part of the cornea and anterior part of the iris and lens. There is posterior chamber, which is covering the space in between the processes of the thin ligament and the iris and lens. There is the chamber of the biggest chamber of the eye bulb, which is covered with the corpus vitreum. Here is the optic nerve. Here is the artery, central artery and central vein of retina. And hyaloid canal, which is the remnant of the embryonic vascular system of the eyeball. Terminology, here is the terminology which I showed you. This is the terms which I use for the description of the eyeball. And it is repeating the terms which I used for description of the eyeball on previous slide. The diagram of the eye showing you the iridocorneal angle with anterior chamber, posterior chamber, corpus ciliaris muscle of corpus ciliaris, ciliary processes, zonula, fibrillin fibers of zonula being attached in between the ciliary processes and the lens capsule, pupil, which is the apertura through which the light rays are entering the corpus vitreum and focusing in the retina. So this is the matter of choice, which slide you will prefer to describe, but all these terms are repeated in the previous slide and on the current slide. The microanatomy of the fetal eye, showing you the simplified version of the histological specimen. Cornea, sclera, corpus ciliaris, ciliary processes, iris, lens, anterior chamber, posterior chamber, and sclera, choroidea, retina, optic nerve, and corpus vitreum. Cornea is transparent, bulging anterior 1/6 of the eye. Anterior epithelium, which is composed of the stratified squamous non-keratinized epithelium with free end nerve endings, responsible for corneal reflex. Thick Bowman's membrane, connective tissue lamina propria of stroma, which is transparent. The cement membrane and posterior epithelium of corneal endothelium, it is otherwise called. Sclera consisting of white opaque sclera covering the fifth sixth of the orb. Sclera is composed of type 1 collagen fibers interlaced with elastic fibers. It is enveloped within the tenon capsule, and thin layer of loose connective tissue between the sclera and capsule of tenon is called episclera. Here the small magnification and high magnification of the cornea, stratified squamous non-keratinized epithelium of anterior surface, the Bowman's membrane, the parallel arrangement of the collagen fibers within the stroma of the cornea, transparent stroma, which is consisting these violet lines are the cells of fibroblasts, of fibrocytes. The cement membrane, here it is visible better on high magnification membrane of the cement, and endothelium of anterior chamber or endothelium of posterior surface of the cornea. Diagram of the eye anterior segment consisting of ciliary body, corpus ciliaris, with ciliary muscle, iris, process of ciliary processes, the thin ligamentozonular fibers composed of the fibrillin, firm fibers, the anterior chamber, the pupil, the lens, the processes of the ciliary processes are having blood capillaries covered with epithelium, which is filtrating the blood plasma, forming the aqueous humor of the eye, responsible for the keeping the pressure, intraocular eye pressure. From the posterior chamber, the aqueous humor is transported, moving through the pupilla to the anterior chamber, and from anterior chamber to the scleral venous sinus or the Schlemm canal, which is located in the limb, in the borderland between the cornea and the sclera, and which is delivering the excess of the watery fluid with the aqueous humor of the eye to the venous system. Here you have the anterior segment of the eye, and another representation, the schematic view of the angle of the cornea, iris or iris corneal angle, with anterior chamber, with posterior chamber, this lens, light microscopic representation, the iris playing the role of the diaphragm, which is changing its orifice through pupilla, depending on the intensity of the light which is entering the eye bulb. Here is the cornea. Here is the process of ciliary body, ciliary processes. Here you have the components of the iris, consisting the anterior layer composed of the fibroblasts-like cells of the connective tissue origin, the stroma, connective tissue stroma, the musculus constrictor pupillae, which is located in radial direction, and musculus sphincter pupillae, which is covering the edge of the iris, responsible for the diminishing the size of the pupilla. Melanocytes are numerous in the posterior surface of the iris, and this is depending on the amount and the quantity of melanocytes is belonging to the color of eye will be dependent. Iridocorneal angle. The iris stroma contains melanocytes and posteriorly has smooth muscle cells of the sphincter pupillae muscle and the myoepithelial cells forming the dilator pupillae muscle. The lens is the unique vascular structure composed of long lens fibers covered on its anterior side by the cuboidal lens epithelium and surrounded by a thick acellular layer called the lens capsule. The lens is suspended behind the iris and its central pupil by the ciliary zonule of fibrillin fibers produced by epithelial cells covering the encircling ciliary body or corpus ciliaris. Here we have another view of iridocorneal angle, anterior chamber, posterior chamber, melanocytes in the iris, the cornea, canal of Schlemm, ciliary muscle. Here will be sclera, ciliary process, also covered with melanocytes and secreting, producing the aqueous humor from its microcirculatory bed, which is filling the posterior chamber, moving to the anterior chamber and moving to the canal of Schlemm when it is in the excess. Here is the trabecular meshwork through which the aqueous humor is moving through the canal of Schlemm. Dense connective tissue of sclera, corneoscleral junction is here. The canal of Schlemm is located in the limbus of the eye. Here again the corneoscleral junction, showing you the trabecular meshwork of the orifice of the scleral vein of sinus, the anterior chamber, the iris, the pigment cell layer, the corneoscleral junction, and ciliary body with ciliary muscle. Here you have the trabecular meshwork of the interface between the anterior chamber and the scleral vein of sinus. Iris, the colored anterior extension of the choroid, is a contractile diaphragm that controls the pupillary aperture. Its structure, anterior surface covered by an incomplete layer of fibroblasts and melanocytes. Connective tissue stroma enriched with fibroblasts and melanocytes. Dilator and sphincter pupillae muscles. Posterior surface is smooth, covered by heavily pigmented melanocytes. Here you have the iris with these components which I announced to you. the anterior surface, the stroma, the musculus sphincter pupillae, musculus dilatator pupillae. Here is dilatator, here is sphincter pupillae, and pigment epithelium and posterior chamber with remnants of the lens capsule. This is the pupilla, pupil. Ciliary muscle contraction has responsibility for the process called accommodation. Contraction of ciliary muscle makes lens more convex, enabling focusing the eye on the nearby objects. Ciliary processes, about 70 in total, radiate from the central connective tissue cord over the lens. They produce aqueous humor, which is responsible for the maintenance of intraocular pressure. The excess of the intraocular pressure is causing the problems with the nutritional support of the retina. It's called glaucoma. This light-threatening or visual-threatening conditions. Aqueous humor from posterior chamber flows through the pupil into the anterior chamber and is drained by the scleral venous sinus or Schlemm canal located in the corneoscleral junction, which is called the limbus of the eye, into the venous system. Sonular fibers radiating from the ciliary processes to insert into the lens capsule regulate the convexity of the lens. Here you have the Schlemm canal. Syn ligament or the suspentacular ligament, the ciliary processes with melanocytes, anterior chamber, posterior chamber, fragment of the lens, pigment epithelium on the iris, which, as I told you, is responsible for the color of the eyes. The more melanocytes, the more dense black or brown color of the eyes will be visible. The ciliary muscle, connective tissue stroma of the ciliary processes. Ciliary muscle and processes, ciliary muscle, ciliary processes, syn ligament, anterior chamber, posterior chamber, iris, scleral venous sinus of Schlemm canal, the conjunctiva, corneoscleral junction, and blood capillary network of the ciliary processes, which is covered with cuboidal epithelium, and it's from the blood of the ciliary processes, the aqueous humor is moved to the posterior chamber, and from that to the anterior chamber through the pupil and to this canal of Schlemm. This is the histological specimen of the lab guide, which I show to you in order to be capable of making the designated remarks. Here is the artifact. The iris is attached to the cornea. That is why we don't see the anterior chamber space. This is the artifact which has developed due to some problems with sectioning material and not removing the iris from the cornea in the sectioned slides. Here you have the corpus ciliaris, the ciliary muscle, the retina, the choroid, sclera, lens, syn ligament, fibers. And here you have the cornea to which the iris has been attached as the result of improper sectioning material. Here is the choroid with attached retina. The choroid with microcirculatory bed, including the bigger size vessels in the intermediate layer of choroid, including the blood capillary or choroid capillary, choriocapillary layer contacting the pigment epithelium of the retina. And here you have the photoreceptor outer segments of the rods and cones, containing rods and cones of the photoreceptor, bipolar neurons, pericardions of which are located in the external granular layer. Here is the schematic view of the circulatory bed. And here you have the ophthalmoscopic view of the retinal blood vessels, taking origin from the optic nerve. And here is the fovea centralis, the better spot of the optimal view for each particular eye. You have choroid and photoreceptors outer segments on the light micrographic representation, choroid, choriocapillary layer of choroid, tunica vasculosa, pigment cell layers, pigment epithelium layer, photoreceptor, outer segments, including cones and rods. Here you have this electromicrographic representation of the same part of the eye bulb, the sclera, choroid, choriocapillary network, and the outer segments of the photoreceptors of the retina. Retina pigmented epithelium with granules of the melanin in the cytoplasm. Lens, the diagram of the lens and the light microscopic view of the lens with capsula, with epithelial cells covering the surface of the posterior part of the lens. Here you have the anatomy of the lens, showing you the lens fibers, showing you the surface of the lens covered with epithelium, showing you the pupil, iris, anterior chamber, posterior chamber, ciliary muscle, the process of this ciliary body are attached to the capsule of the lens, and lens is changing the convex surface, changing the shape due to the attachment of the contraction of the ciliary muscle and attachment of the zonular fibers of the thin ligament. These lens fibers are nothing else as the modified cells of the lens, filled with the protein and having the transparent composition of the, changing the direction of the light rays and focusing them on the retina, on the optic part of the retina of the posterior part of the eye bulb. Here you have developing lenses, cunning electron microscopy of the major lens fibers. Posterior lens capsula, the embryonal nucleus of the lens developing from the lens placodes on the surface of the embryo, ectodermal structure of the head region, the fetal nucleus, and the anterior lens capsule, and lens fibers filled with the crystalline protein which is having the responsibility for transparency of the lens ultrastructure. Here the scanning electron microscopy of the ciliary body, iris, pupil, and zonular fibers from behind of the view from behind. This is the pars plana of the ciliary body, pars plicata of the ciliary body, the iris from the posterior view, the pupil from the posterior view, and thin fibers of the thin zonula or the thin ligament connecting the process of the ciliary body to the lens capsule, modulating the shape of the lens. The more contracted will be ciliary muscle fibers, ciliary muscle cells, smooth muscle cells, the more globular will be shape of the lens, and it is the particular condition under which the located in nearby objects will be distinctly visible. When the lens muscle is relaxed, then the eye is focused on the distant objects. And this is the condition when the smooth muscle cells of the ciliary muscle are relaxing. Here the scanning electron micrograph of the lens, ciliary processes, and zonular fibers from behind. The lens surface, the zonula of the thin ligament, the ciliary processes, and ciliary muscle on the right scanning electron microscopic magnification. The retina has two major parts derived from the embryonic optic cup. You will see it later on the final segment concerning the structure of the eye and its development. The pigmented epithelium next to the vascular choroid layer and the thickened neural retina. Neural retina is derivative of the neural sites and neural glial cells of the developing cerebral cortex, cerebral part of the central nervous system. Cells of the pigmented epithelium absorb scattered light, form part of the blood-retina barrier, regenerate 11 cystrin retinal, the phagocytose, the shed discs from the rods, and support the rod and cone cells. Rod cells are photoreceptors detecting light intensity with short rod-shaped outer segments. The numerous coneless cells with conical outer segments and receptors for the primary colors. Light of different wavelengths fixed by the cones. Rods have stacked membrane discs in which the membranes are densely packed with the protein rhodopsin with the bound retinal. Photons of light convert 11 cis retinal to all trans retinal, causing rhodopsin to release the retinal bleaching and activate the adjacent G protein transducin, which causes a nerve impulse generation. In the neural retina, the rod and cone layer is nearest to the retinal pigmented epithelium and near the inner nuclear layer, which contains the cell bodies of these photoreceptors. Outer plexiform layer contains the photoreceptors, axons connected in synapses with dendrites of the various integrating neurons whose cell bodies form the inner nuclear layer. Axons from cells in the inner nuclear layer form synapses with the interplexiform layer with dendrites of ganglionic cell layer, which send axons through the nerve fiber layer to the optic nerve. Here you have the diagram of the posterior part of the eye, showing you the optic nerve with fibrous capsular coat, the sclera, choroid, retina, choroid, retina, with pigment epithelium contacting the choriocapillary layer of the choroid, the rods and cones layer, the outer membrane layer, the outer plexiform layer, the inner granular layer, the inner plexiform layer, the ganglionic cell layer, the nerve fibers layer, and the inner ligament, inner membrane contacting the from one surface contacting the neural retina, and from the other surface contacting the corpus, the vitreous corpus vitreum. Here you have the impulse response to light through the retina and incoming light with blue color, and impulse response to the light through the retina coming. Here are the sclera with choroidea and with optic retina and complete disposition. The pigment epithelium, here is the retina. Letter R is encompassing the retina. The pigment epithelium, the rods and cones layer, the outer limiting membrane, the outer nuclear layer or granular cell layer, the outer plexiform layer, the inner granular layer, the inner plexiform layer, the ganglionic cell layer, the nerve fibers layer, and the inner limiting membrane is delineating, bordering between the corpus vitreum and the optic retina. Here you have the cellular composition and the corresponding layers of the optic retina with terminology which is used for its description. The choroid, the pigment cell layer, the photoreceptor layer of rods and cones, the outer limiting membrane composed of the Muller gliocytes or Muller glial cells. Here they are, the Muller glial cells. The cell bodies of rods and cones, the perinuclear regions of the rods and cones containing cells. The outer plexiform layer, where the synaptic junction between the axon of the first neuron, photosensory neuron, and dendrite of the second bipolar neuron. Outer plexiform layer is consisting of it. Inner nuclear layer consisting of pericardions of the bipolar new associating neurons of the three neuronal chain of retina, and with pericardions of the amacrine cells and the horizontal cells, which are also neural cells, which are delineating the impulse generated by the photosensory rods and cones, delivering these impulses to the within the lateral disposition. The inner plexiform layer consisting of the synaptic junctions between the second neuron and the third neuron of the retina, the ganglionic cell layer, the optic nerve fibers layer, and the inner limiting membrane. Corpus vitreum, vitreous body. Here is the cellular composition of the retina, consisting of the pigment epithelium with melanin granules incorporated to the processes and to the cytoplasm of the pigment cells, the rod, the outer segment of the dendrites of the first bipolar photosensitive neuron, the cones, the pericardion of the photosensory bipolar neurons, the horizontal cells, the amacrine cell, the nucleus of the second neuron, bipolar neuron, the nucleus of the Muller cell, the inner plexiform layer consisting of the synaptic junctions between the second associating neuron and amacrine cells, distributing the action potential in the lateral direction, and the ganglionic cell pericardia, the nerve fibers layer, and the inner limiting membrane formed by the terminal portions of the Muller cells, both membranes, the outer limiting membrane and inner limiting membrane is the terminal portions of the Muller nucleosides. So here is the denominated the cellular components of the retina of the eye, which I delivered to you on the picture. Here you have the 10 layers of the retina, including pigment epithelium, rods and cone photosensory layer, outer limiting membrane, outer nuclear layer, outer plexiform layer, inner nuclear layer, inner plexiform layer, ganglionic cell layer, optic nerve fibers layer, and inner limiting membrane. I showed it on the picture. Here you have the sclera, choroid membrane, and the retina with 10 layers delineated from the corpus vitreum by the inner limiting membrane or inner limiting layer. And here you have the optic retina with the layers and with choroidea, choriocapillary layer of the choroidea attached to the retina from the outer surface. Here you have the same micrographs, but taken from your lab guide in order to make it convenient to you to make designatory marks for these layers of the posterior surface, posterior part of the eye. And don't make mistake, this is not the retina, this is sclera, this is choroidea, and retina starts from the thin pigmented epithelial layer in between the choroidea and the optic retina. Here is the corpus vitreum with inner limiting membrane between the retina and the corpus vitreum. Here you have the specific areas of the retina. This is the fovea centralis, the best vision zone of the retina, which is having only pigment cell layer, the rod and cones layer, the outer limiting membrane, outer granular layer, and outer plexiform layer. All the rest are removed. And here is the site of the retina where the highest amount of the cone, photosensitive for colored light rays of different wavelengths are located. The most sensitive part of the retina is the fovea centralis, luteal spot of the optic retina. And here is the blind spot, the optic nerve leaving the retina, delivering the light rays to the cortex of the brain, where the images are formed from the signals coming from the retina of the eye. Light-sensitive photoreceptor functions of the retina are attributed to rods and cones, modified dendrites of the retinal photosensory neurons. Rods are sensitive to dim light. Each eye contains about 100 or 120 million rods. Each rod consists of membranous discs into which photosensitive pigment rhodopsin is incorporated. There are 3 types of cones which are sensitive to red, green, and blue light waves. Each eye consists of about 6 million cones and 100 to 120 millions of rods. It's more numerous, about 50 times more numerous are the rods. And there are only 6 million cones into membranous disc of which photopigment iodopsin is incorporated, sensitive for different wavelengths, light rays. And the most of the cones, as I told you, are located in the forward centralis region of the retina. Here you have the light micrographic representation of the outer nuclear, outer granular layer of the retina, consisting of the nuclei of the first photosensitive neuron. Here the dendritic process of the neurons, the cones and the rods are visible. Here is the pigment epithelium processes which are filled with melanin granules. Here is the nucleus of epithelial cells. And here is the structure depicting the rod bearing bipolar neuron, cone bearing bipolar neuron, Mueller cells which are supporting, giving mechanical support, phagocytizing the damaged parts of the outer segments of the photosensory neurons, giving the nutritional support, playing the role of the nurse for the nursing element for the photosensory neurons. And you see the size and form of the outer segments of the photosensory neurons of the rods and cones is different, and different sensitivity to light rays of different wavelength. Here the cone and rod, the diaphragm and the diagram and the electromicrographic representation of the membranous discs of the outer segments of the rod, the connecting stack of the cilium-like arrangement with microtubules inside, the membranous hemidisc of the cone. Here you have the shape and the structure of the external plexiform layer forming the synapses with bipolar neurons of the axons of the bipolar photosensitive neurons with bipolar associating neurons contacts. Metabolic region, the proteins and phospholipid synthesis plus ATP production is in the pericardium and outgrowing segments of the bipolar neuron, photosensitive neuron. And photosensitive region, the generation of the receptor potential due to the light rays hitting the iodopsin or rhodopsin of the rods and cones. Here the membranous discs of the rods electromicrographic representation. Here you have the limiting, outer limiting membrane, and here is the inner segment of the bipolar photosensitive neurons. Mechanism of phototransduction. In the dark conditions, the discs which are including the rhodopsin or iodopsin and retinal is forming the complex structure. Inactive rhodopsin consisting of opsin and retinal. And transducin is accomplishing the one single entity. When the light rays hit this highly structurized complex of the protein of substances, the opsin is dissociating from the retinal. Activated retinal is dissociating from opsin molecule. The transducin is dissociating from the rhodopsin molecule, binding with the phosphodiesterase enzyme, activating the sodium canaliculus through the plasma membrane. It is closed, and the deficit of sodium ions in the cytoplasm of the cones and rod is delivering the action potential, which is further transported to the associating neurons of the optic retina. The eyelids and lacrimal apparatus are the associated structures associated with the eye bulb. The eyelids are lined by conjunctiva, stratified columnar epithelium with goblet cells, which also covers the anterior part of the sclera, and it continues with the corneal epithelium. The lacrimal glands continuously produce the tear film that drains into the nasal cavity via the ducts of the lacrimal apparatus. Here you have the eyelid, conjunctiva, and tarsal glands, secretory units, or meibomian gland secretory units. Here you have this small magnification of the eyelid, the conjunctiva, the stratified columnar epithelium with superficially located goblet cells secreting mucus-rich substance, and meibomian gland, which are modified sebaceous gland, which is covering the space of the open space of the eye, preventing its exsication, preventing its damage. Here you have the red coloration of the blood capillaries, which are showing you inflammation of the eye, which is called conjunctivitis. The surface of the eye, the blood vasculature, microvascular bed is overloaded with blood. Here you have increased magnification, the tarsal gland, the musculus of the skeletal muscle, fibers of the eyelid, the conjunctiva surface of the eyelid, the epidermis, the skin, cutaneous surface of the eyelid, the follicles of the eyelashes, and the tarsal glands, the secretory units, the modified sebocytes secreting due to the holocrine secretion. Here the sagittal section of the eyelid from the left guide, which is supposed to make designated marks, straight skeletal muscle fibers, the cutaneous surface, the conjunctival surface, the tarsal gland, the follicle surrounding the eyelashes. And this is the whole structure of the eyelid. And lacrimal apparatus, the orbital part of the lacrimal gland and palpebral part of the lacrimal gland. Here is the description, the slight magnification, small magnification of the lacrimal glands apparatus. Is increased magnification of the high magnification of the lacrimal gland secretory units. And here is beautiful structure of the mucus-rich secretion of the lacrimal glands secretory units, the new epithelial cells surrounding the duct portions. This beautiful picture is to some extent giving the recollection of the large salivary glands of the mucus secreting sublingual gland of the digestive system. Here the eye development gestation age, beginning of the 4th week and the end of the 4th week, late 4th week, the optic cup developing from the ectoderm. The lens pit is developing from the lens placard, the specific site of the surface ectoderm, the optic stalk, the two-layered of optic cup, the subsequent development of the lens from the lens placard, development of the cornea, developing from the eyelids, developing of the choroidea, developing of the retina, of the neural retina, and the newborn eye of the newborn is consisting of all these components. Its eyelids having capability of opening, and the cornea, iris, lens, corpus vitrum, everything is already developed but not fully matured. Ophthalmological pathologies, which I recommend to you to seek in the textbooks or to seek in the internet and compare with these normal conditions, normal histophysiological conditions, which we described, which I delivered to you today on this lecture. And the second half of the lecture will be dedicated to the vestibular auditory system, which is consisting of the outer or external ear, the middle ear, and the internal or inner ear. The external ear is consisting of the auricle, the external acoustic meatus, and the eardrum, tympanic membrane. Here you have the middle ear consisting of the ossicles, malleus, incus, and stapes. The Eustachian tube consisting the tubular arrangement from the middle ear to the nasopharynx, and oval window, which is housing the part of the stapes, which is moving with response to the movements to the wavelengths of the outer air, and reflecting these vibrations of the tympanic membrane to the inner ear, internal ear, which is consisting of the cochlea, vestibulum, and semicircular canals. And it's hidden in the petrous part of the temporal bone. Tissues of the ear mediate the senses of equilibrium and hearing, having attributed for this purpose mechanisms of mechanoelectrical transduction. The acoustic meatus of the external ear ends at the tympanic membrane, and its mucosal contains sebaceous and ceruminous glands that produce an antimicrobial substance, the cerumen. The tympanic cavity of the middle ear opens to nasopharynx via the auditory or Eustachian tube. Within the tympanic cavity, there are articulated series of three small bony ossicles. The malleus, incus, and stapes connects the tympanic membrane to the oval window in the wall of the inner ear. The inner ear consists of the membranous labyrinth containing endolymph. The membranous labyrinth is enclosed in the temporal bone, bony labyrinth, which contains perilymph. Here, this similar to the previous transparency, the outer ear, the middle ear, and inner ear. Just I take this picture because it's in your lab guide without designatory marks, so you can use this picture to make designations to all these structures which are depicted on the. These are the terminology which are used, terms which are used for the description of the ear and vestibular auditory system as the whole. The external acoustic meatus, ceruminous and sebaceous glands. Sebaceous glands, ceruminal glands, the surface of the stratified squamous epithelium, which is similar to what we have in the epidermis, the epidermal epithelium, and connective tissue corvates incorporated all these glands. The ossicles and the auditory tube, mucus secreting glands or seromucous glands with epithelium simple columnar epithelium, and elastic cartilage, which is making the background, the supporting mechanical supporting the middle ear, especially in the Eustachian tube. Diagram of the bony and membranous labyrinths. Semicircular canals are housing semicircular ducts, which are represented by the pink coloration, 3 semicircular ducts with sensory areas consisting the crista ambularis sensitive zones. The vestibulum, which is consisting of the sacculus and utriculus, sac-like structures with otolithic macula sacculi and macula utriculi covered with otolithic membrane, and ductus cochlearis incorporated to the cochlea of the inner ear. And you see here the membranous labyrinth of cochlear duct in the central position. In the upper part of the bony labyrinth is located the vestibular part and the tympanic part of the bone labyrinth of the cochlea. This is a diagram of bony membranous labyrinth, the sensory elements of the inner ear. The bony labyrinth includes the cochlea, the vestibulum, and the semicircular canals. The membranous labyrinth consists of the cochlear duct, sacculus and utriculus, and 3 semicircular ducts. And the sensory areas are the crista ampullaris, macula sacculi and utriculi, and spiral organ of Corti in the subsequent membranous components of the inner labyrinth. Macula sacculi and utriculi. The membranous labyrinth has a central vestibule with 2 subdivisions. The utricle connects to the 3 semicircular ducts and the saccule connects to the cochlear duct. The walls of the utricle and saccule each have a thickened area, the macula, which contains both sensory hair cells with synaptic connections to sensory nerves and supporting cells. A bundle of rigid stereocilia and 1 rigid kinocilium project apically from each hair cell and are surrounded by the endolymph containing a gel-like matrix with mineralized crystals called the otoliths, so otoconia, calcium phosphate crystals. Head movements cause endolymph and otolithic membrane to move, deforming the rigid apical structures of the hair cells, depolarizing them and producing nerve impulses. Here the macula of utricula, and this is the otolithic membrane with otoliths, endolymph covering the sac of the utriculus and sacculus. The type 1 and type 2 hair cells, otoliths, and terminals of the dendrites of the photosensor, sensitive to the movements of the hair processes, the dendritic terminals or the nerve terminals, taking the electric stimulus, electric charge, which is produced by the hair cells with respect to the movements of the head. Here you have the type 1 hair cell, which is pear-shaped with kinocilium and stereocilia contacting the otoliths within the otolithic membrane, and type 2 hair cell with afferent nerve endings, also containing the kinocilium and stereocilia. Here's it's in diagram of the type 1 and type 2 hair cells with the different shape of the nerve terminals with stereocilia and kinocilium, And it's in neutral position, the fixed vertical position of the stereocilia with respect to kinocilium. Here you have the side links of between the and tip links between the stereocilia and side links between the stereocilium and kinocilium with mechanoelectrical transition channels responding to the movement of the stereocilia with respect to the kinocilium. In this left-hand corner, we have the mechanical deformation of towards the kinocilium, opens the potassium channels in the stereocilia. And this is leading to the influx of the calcium ions through the calcium sensitive channels and the action potential elaborated by the hair cells in which the stereocilia are moving towards the kinocilium. They're taken by the afferent axon terminal, moving to the specific ganglion, the ganglion which is consisting the nerve pericardium of the nerve cell bodies. And the movement of the stereocilia in the opposite direction from kinocilium is closing the calcium and potassium canals, and the closure of the canals, mechanoelectrical canals, is causing hyperpolarization and the negative impact on the synaptic junction with the afferent axon terminal. Here's the ultrastructure of the scanning electromicrographic representation of the stereocilia with kinocilium, with the otoconia and otolytic membrane or otoliths. Otoliths in the otoconia and the increased magnification of the three-dimensional scanning electromicrograph representation. Here you have the macula, sacula, and utriculi with another illustration, kinocilium, stereocilia, and depending on this side to which the stereocilia will be moving, either to kinocilium or to the opposite direction, the electric signal is elaborated or it is blocked, and the plus minus signal is taken by the axon terminals, delivering into the cochlea, vestibulocochlear ganglion. Cristam polaris and cochlear duct. Each of the semicircular ducts oriented 90 degrees from the one end to another has a terminal ampulla region with a thickened cristam polaris containing hair cells that contact the gel-like cupola. Head movements displayed endolymph and stereocilia of hair cells in the utricle, sacula, and semicircular ducts, which together produce signals that contribute to the sense of equilibrium. The cochlear duct is the middle compartment, scala media, of the cochlea and runs between the two other long compartments that contain the perilymph, the scala vestibuli and scala tympani. Here you have the cristam polaris, consisting the type 1 and type 2 hair cells, or they are otherwise called vestibulocytes, based on the supporting cells. And the cristam polaris is covered with the cupula gelatinosa, which is surrounded with the endolymph, and turning the cupula gelatinosa is causing the drift, the movement of the stereocilia with respect to kinocilium or in the opposite direction from the kinocilium to stereocilia. This is making the same signal of negative or a positive or negative signal from the hair cells surrounded with the axon terminals of the sensitive neuron of the vestibular cochlear ganglion. Here is the cupula gelatinosa. Here is the crista ampularis, hair cells type 1 and type 2. The signal is delivered to vestibular branch of the cranial nerve of the 8th vestibular cochlear nerve ganglion. Ampulla cupula, vestibular branch of the nerve, cranial nerve 8. Here is the light micrographic representation of both crista ampularis with cupula on the surface and the macula, saculi and macula utriculi with otoliths, otoconia on the apical surface. Reacting the crista ampularis to the angular acceleration and the macula, saculi and utriculi to the linear acceleration of the gravitation signals, changing the positioning in the of the head or the whole body with respect to the ground, with respect to the vertical lines responding to gravitation. Spiral organ of Corti. Along the base of the cochlear duct, the basilar membrane supports the spiral organ of Corti, which consists largely of the hair cells connected to sensory fibers to cranial nerve 8. The cochlear hair cells include 3 to 5 rows of outer hair cells with stereocilia embedded in a jet-like tectorial membrane and 1 row of more heavily innervated inner hair cells. Sound waves transmitted by the ossicles move to the oval window and produce pressure waves in the cochlear perilimb, which deflects the basilar membrane, an organ of the Corti, causing nerve impulses which the brain interprets as sounds. Here the cochlear duct with scala vestibuli, scala tympani, ductus cochlearis, scala vestibuli, scala tympani, ductus cochlearis. The spiral organ of Corti, inner tunnel, outer cells of the spiral organ of Corti, inner cells of the spiral organ of Corti, and the cochlear branch of the cranial nerve 8 is depicted with yellow coloration. This is tectorial membrane, which is vibrating with respect to the vibrations of the air taken by the tympanic membrane. Here is another high magnification of the spiral organ of Corti with its location within the scala media, with the tectorial membrane covering the spiral organ of Corti, with the outer hair cells, with the outer phalangeal cells, outer pillar cells, outer Hansen cells. These are supporting cells which are having an abundance of names. Cells of Claudius, cells of Belcher, heads of Hansen, and cell of Daters. It's all supporting cells of the outer supporting cells of the spiral organ of Corti. These are inner cells, which are located between the inner spiral cells, in between the inner phalangeal cells and the inner hair cells covered with tectorial membrane. And here you must know this is vestibular membrane of the Reisner membrane. This is the spiral prominence. This is the stria vascularis, ligamentum spiralis, scala tympani, scala vestibuli, Reisner membrane, and scala media, tectorial membrane. And these vibrations of the both perilymph, covering the space between scala vestibule and scala tympani, endolymph, covering the space between the membrane, Reissner membrane and spiral organ of Corti. Its vibrations with respect to the eardrum, to the external membrane, covering the interface between the external meatus acousticus and middle ear, and with beating the moving of the ossicles of the malleus, incus, and stapes. These vibrations are transmitted to the perilymph, and these vibrations of perilymph are transmitted to the vibrations of the spiral organ of Corti basilar membrane, on which are located the spiral organ of Corti. Here is basement membrane or basilar membrane and dictorial membrane. These vibrations are topographically specific, and this topography of the hair cells, which are stimulated by the vibrations, are transmitted to the cochlear, vestibular cochlear ganglion, to the vestibular cochlear nerve. Reacting with the generation of stimulus or action potential. Here you have the axial section of the cochlea with 5 cavities: ductus cochlearis, scala vestibuli, scala tympani, spiral organ of Corti, the spiral organ of Corti. Enlarged magnification of the axial section of the cochlea, which is in your lab guide, which you are supposed to be capable of depicting the designatory marks. Here you have the increased magnification of the one single ductus cochlearis surrounded by scala vestibuli and scala tympani with spiral organ of Corti and the basilar membrane, and the limbus of the inner ear and vestibular membrane of Reissner, delimiting the scala vestibuli from ductus cochlearis, stria vascularis, and ligamentum spiralis are the outermost structures. Here you have the spiral ganglion. Here is increased magnification. This is Reissner membrane of vestibular membrane, scala vestibuli, scala tympani, basilar membrane, spiral organ of Corti, tectorial membrane. The limbus of the inner ear, the stria vascularis, ligamentum spiralis. Stria vascularis is serving for the secretion of the fluid, which is forming the endolymph, filling the space of the membranous labyrinth of the inner ear. Here is the spiral organ of Corti with the limbus, or spiral limbus, vestibular membrane of the Reissner, tectorial membrane, inner hair cell, inner pinnar cell, inner pillar cell, inner phalangeal cells, inner tunnel, outer phalangeal cells, outer hair cells, and outer supporting cells and basilar membrane visible on the basement part of the spiral organ of Corti. Here is the increased magnification of your lab guide. Again, without designation, you're supposed to make it by your own designatory marks for the description of this picture. Here you have the stereocilia of outer hair cells under scanning and electron microscopic magnification. Here is the histophysiology of sound perception. The movement of the stapes covering the oval window is transmitted to the scala vestibuli, then through the helicotrema, moving to the scala tympani, moving to the round window, and the movements of perilymph is causing the movements, the vibrations of the varicose membrane, vibrations of the basilar membrane, vibrations of the spiral organ of Corti, with response to the quantity evaluation of the light of the sound waves. So you see here the different segments of the spiral organ of Corti. Hair cells are responding to the low frequency sounds at the end of the spiral organ of Corti, of the very end mount. Medium frequency sounds are giving vibration to the middle part of the spiral organ of Corti, and high frequency sounds moving, giving signal to the part of the spiral organ of Corti close to the oval window. Here you air development on the 4th week, where the first pharyngeal membrane is transformed to the air drum, where the first pharyngeal pouch is giving birth to the proximal pharynx and the auditory tube, and the middle ear. The otic vesicles, including the otic placode, is transformed to the vesicular structures and transformed to the membranous labyrinth in the inner ear. Here you have the otic vesicles transformation, forming the membranous labyrinth of the inner ear. The membrane of the first pharyngeal pouch is giving birth to the middle ear and ossicles, and the external acousticus, meatus acousticus, is delivered from the first pharyngeal membranes. It's again a lot of terminology which you're supposed to be capable of using for the description of both the adult structure of the ear and embryonal steps of the embryological development of the inner ear. Pathologies related to the ear: otitis media, vertigo, dizziness, motion sickness, many ear disease, vestibular neuritis, conductive hearing loss, and sensory neural deafness, and benign proximal positional vertigo. You can find this description in the textbook, or you can find it in the internet, and compare. I recommend you to compare with the normal conditions which described just now at the lecture. Sense of taste and smell. The dorsal surface mucosa, this is just several final the transparencies I'm going to describe to you. The dorsal surface mucosa of the tongue has projecting lingual papillae of 4 types: filiform papillae with keratinized epithelium and non-keratinized foliate, fungiform, and large vallate papillae. All lingual papillae except the filiform type have epithelial taste buds on their sides, which have protectory gustatory cells with synapses to basal sensory innervation, support cells, basal cells, and apical taste pore. The roof and part of the superior concha in each nasal cavity are covered by olfactory epithelium, which is pseudostratified epithelium containing bipolar olfactory neurons, support cells, and stem cells. So, the eye and the olfactory epithelium are consisting of the neurocytes, which are sensitive cells of this sense of seeing and smelling, and the cells of the hearing and gustatory perception is derived by the specific epithelial cells, which are specifically developing to catch this stimuli from the outer space and transport to the nervous system to nerve terminals. Here the taste buds of the tongues and sense of taste receptors located in the lateral position of the circumvallate papilla. Here you have this green coloration showing the lateral position of the fungiform papilla and circumvallate papilla. Here you have the slight micrographic representation of the taste buds with taste spore and receptor, gustatory receptor cells supported with the basal cells and supported with the supporting sustentaculi cells. Here is the epithelium of the surrounding epithelium of the dorsal part of the tongue. And there is the olfactory region of the nasal cavity. You see its location. You see these three neuronal components, the sensitive nerve olfactory receptor cell, the basal cell and sustentacular cells, which nuclei located on different levels. That is why the cells are, the epithelium is treated as pseudostratified epithelium. They have the Bowman's gland, while in the sense of the gustatory perception, there are the von Ebner glands, which are secreting similarly to the Bowman's glands. Here you have this sustentacular cells. And here you have olfactory cilia, which are responsible, to which are incorporated the sensitive proteins, sensitive to different tastes, which are taking the stimuli of the taste of the food and drink, which we are keeping in our mouth, in our oral cavity. And the very last segment is showing you the recommended reading for the topic, the organs of special senses. Thank you for your attention. It's all what I pretend to describe to you. Have a nice day.